My presentation is about the amelioration of non-alcoholic fatty liver disease by hepatic stimulator substance via preservation of carnitine palmitoyl transferase 1 activity. So before I start, I want to dissect the title of the paper. I'm going to introduce these three topics in my introduction, and I want to show the effects of non-alcoholic fatty liver disease and what it actually is, the importance of the hepatic stimulator substance and what it actually is as well, and the CPT1. And all in all, I want to introduce how all three concepts relate to one another. Non-alcoholic fatty liver disease is when there's a buildup of fat in the liver. It's characterized by steatosis, which is when fat is deposited into the liver. So non-alcoholic fatty liver disease is caused from other causes than alcohol use. So it's found to have a 30% prevalence in the United States. So it's characterized by insulin resistance and metabolic syndrome. Symptoms such as fatigue, malaise, weight loss, and mild jaundice are typical for this disease. So the disorder ranges from simple steatosis to non-alcoholic steatohepatitis. It's similar to a progression of like stages, just like cancer. So they begin to progress to cirrhosis, which is the last and worst stage. This includes liver failure and the accumulation of carcinoma in the non-alcoholic steatohepatitis, or NASH for short, is characterized by steatosis, inflammation, and the infiltration of inflammatory cells. So, going back to what NAFLD was, I mentioned how it could range from simple steatosis to NASH, and then to cirrhosis. So it is characterized by the steatosis inflammation, which is an accumulation of triglycerides. It works in a two-hit mechanism. So how this process works is that the first hit in the mechanism is associated with obesity and a metabolic disorder due to the liver steatosis. Lipid accumulation in the liver occurs due to a number of reasons. Due to the excess of free fatty acids from high-fat diets, release from adipose tissue, or an increase in hepatic fatty acid synthesis, and decrease in the degradation. So the second hit is a multifactorial process, which involves apoptosis and necrosis, as well as a massive production of reactive oxidative species, such as peroxides, hydroxyl radicals, singlet oxygen, and these can potentially lead to cell damage. Since is a liver protective factor, hepatic is of or relating to the liver. The importance of the hepatic stimulant substance is that it promotes hepatocyte proliferation and liver regeneration after a specific procedure called hepectomy. So this would protect the liver from acute injury from the radical oxidative species that I talked about previously. So theoretically, it's proven to protect mitochondria from mitochondrial permeability transition. Palmitoyl transferase 1 acts as a mitochondrial gateway for fatty acids to enter within the matrix. It's shown to induce mitochondrial gene expression and enhance oxidative phosphorylation within the liver mitochondria. So it protects mitochondria and acts as an inhibitor, which would prevent cell damage. So basically, what CPT1 is an essential enzyme that's responsible for beta oxidation of free fatty acids in mitochondria. It's also in charge of the formation of acyl carnitines. So the purpose of this paper was basically to find whether HSS, protection of the hepatic mitochondria, is linked to mitochondrial metabolism or the activity of CPT1. Also, whether CPT1 could be one of the effector molecules for HSS protection. So they're basically trying to find whether HSS would protect carnitine palmitoyl transferase from lipotoxicity, which would alleviate hepatic lipid disposition. So what lipotoxicity is it's a syndrome that results from the accumulation of lipids in a tissue. Injecting the HSS gene into male A57BL6J, which were between 18 to 20 grams. So these rodents would be fed a specific diet called methionine choline deficient diet, which would develop into steatohepatitis, which was to mimic patients with NASH. So these rodents would be fed with high fats for 16 weeks. In total, there were 120 mice that were used. 30 mice were the control group, which were fed normal chow diet, and the remaining 90 would be fed the actual methionine choline deficient diet. So these, as well as the diet, these Hep G2 cells were transfected with the HSS plasmid and oleic acid treatment. Experiment, we wanted to see if the HSS gene was able to protect the liver cells from MCD-OA-induced steatosis, which would most likely lead to the regulation of CPT1 activity, as well as help with the CPT1 activity to decrease. So figure 2A is a bit confusing, but I'll take you through it. In the x-axis, you see time measured by weeks up to the 4th week. On the y-axis, you see the body weight measured by grams. So the body weight of the mice that were fed the control weight rose to 28 grams. So this makes sense in terms of the actual disease and what it does. So moving on, the body weight of mice with the HSS gene transfection, the non-transfected mice, and vector transfected mice, these three groups showed a clear decrease in body weight. However, the HSS transfected mice showed that HSS transfected mice was able to improve the MCD diet-induced body weight loss. Was a qualitative representation of how hepatic steatosis was more serious in the livers of MCD-fed mice compared with the livers of mice that underwent HSS gene transfection after the diet. So there was more lipid accumulation in the liver tissue from the non-transfected or vector transfected mice compared to the control or HSS transfected mice. So this can be seen by the deep red color of the oil red O staining. The F480 staining detects macrophages, which detects hepatic inflammation by measuring the mRNA levels of inflammation factors. If you see under the MCD group and the MCD and vector TX group, you see these large bubbles, which is an indication of hepatic inflammation. And H&E staining is just a typical way of visualizing the actual tissues. How HSS increased the mRNA and protein expression levels of CPT1 in HPG2 cells. So this was shown to see if the HSS transfected cells was able to help cells increase the metabolism of fatty acids in mitochondria. So if you see in graphs B and C, it showed how oleic acid treatment did not help CPT1 expression, but there was a substantial increase in the HSS expressing cells. Starting on the graph on the left, it showed the different levels of expression between the control oleic acid treatment, oleic acid treatment and vector TX, and oleic acid treatment with HSSTX. The second graph, right next to it, showed the comparison between the control oleic acid treatment, acid treatment, and vector TX, and oleic acid treatment and HSS RNAi. The graphs were separate in order to show the comparisons between the maximum and minimum treatments. Graph C shows the mRNA expressions, which show that HSS transfected treatments would have the greatest level of expression, and OA-HSS RNAi the lowest. This meant that this would help the cells increase the metabolism of fatty acids, which was shown in figure 9D with the oil red O staining, which had a substantial less amount of fatty acids present compared to the other groups. However, in contrast, there was a decrease in the HSS siRNA cells, which showed the largest accumulation of lipids in the cytosol. This was implicit of reduced CPT1 activity. So this paper's focus was about how non-alcoholic fatty liver disease, or the progressive form NASH, operates in model organisms within the research, and how that would translate to NASH in humans. So The focus was to determine therapeutic methods for interventions that would help patients with NASH. So, leading to that, they found that the hepatic stimulator substance would help to promote liver protection and regeneration. But there's still yet to be um, a lot of extensive research on the substance, specifically whether it could rescue these hepatocytes from lipotoxicity and eliminate excessive deposition in the liver. And it was also a way of investigating how CPT1 activity is regulating fatty acid metabolism in the mitochondrial membrane.